Hello, everyone. My name is Dongyuan Song. I'm a PhD student in bioinformatics from UCLA. Today, I'm going to talk about scPNMF, sparse gene encoding of single cells to facilitate gene selection for targeted gene profiling. This is a collaborative work with Ke-Xin Li and is under the supervision of Dr. Jingyi Jessica Li. Here's the introduction. So as you may know, single-cell RNA sequencing dataset can capture the whole transcriptome. For example, it can measure thousands or 10,000 genes simultaneously. So given single-cell RNA-seq can measure all the genes, why do we still want to select a subset of genes? The first reason is for the interpretability. So compared to 10,000 of genes, a few hundred genes are much easier to interpret. The second reason is denoising. Some of the genes, their variation are not related to your biological interest, and you can treat them as a noise of your dataset. Therefore, an efficient gene selection can help you denoise your dataset. The third reason, and also our focus, is the targeted gene profiling. Although single-cell RNA-seq is the most popular one, there are still many other methods which are targeted gene profiling methods. For example, RT-PCR, assay targeted gene sequencing, and spatial genomics like MERFISH. Usually, these methods can only measure 10 to 200 genes. Although there are a lot of different gene selection methods currently, most of them tend to select a lot of genes. For example, the default method of Seurat is 200,000. Therefore, those gene selection methods are not quite suitable for targeted gene profiling design. To solve this problem, here we propose the scPNMF algorithm. Briefly speaking, scPNMF tries to find a sparse projection of the expression matrix, and we call this sparse gene encoding. And then you can select the informative genes by this sparse projection. It has very broad applications. For example, it can be used for informative gene selection. It can also help you map your new cells to the reference dataset. And also itself can serve as an interpretable dimensionality reduction method. Here is the method part. And the foundation of scPNMF is the PNMF algorithm. And the PNMF is short for projective non-negative matrix factorization. This method was first proposed in 2009. And based on this name, you can easily think that it is a hybridization of PCA and NMF. So the objective function is like this, where X is the expression matrix with P genes and N cells. And W is the weight matrix we try to estimate with only K bases. And here we want to minimize the forbiddiness of X minus WW transpose X. If we compare this objective function with PCA, we can see they are very similar to each other. The difference is that PNMF, it requires the W to be non-negative. If we compare it with NMF, we can see they are also very similar to each other. But in NMF, you need to estimate W and H together. But in PNMF, you only need to estimate W. Therefore, PNMF theoretically is a combination of PCA and NMF. And this makes it has much higher sparsity and better mutual exclusiveness compared both PCA and NMF. In the first step of scPNMF, we perform the PNMF algorithm and try to estimate the weight matrix W. Here, W has K different bases, and each basis, if we use the terminology from NMF, we can call this metagene. So it represents the weights of a small set of genes, which are co-expressed and potentially functionally related to each other. And the score matrix is a low dimension using W to project X onto the K dimensional space. And here you can see each score vector in S is corresponding to its own basis vector from the weight matrix. In the second step, we want to perform the basis selection. So although we get K different bases, or you can treat this as k different metagenes or k different factors from the PNMF algorithm. Not all of them are related to your biological interest. So here we have the optional step. If you have the prior knowledge, you can perform the functional annotation, for example, Go annotation, and you can remove those factors which are not related to your interest. Of course, if you do not have such kind of prior knowledge, we have two purely data-driven methods. The first one is Pearson correlation with cell library size. So in single cell analysis, people usually treat cell library size as a confounding factor. Therefore, if one score factor is highly correlated with the cell library size, we can remove this since we think that its variation is not related to the biology. And the second approach is a multimodality test. The basic assumption of this is that if a score vector shows very clear multimodal pattern, it must represent some discrete heterogeneity structure of your underlying cell population. Therefore, we can perform the multimodality test and only keep those bases with significant p-values. After this basis selection, we got the selected basis weight matrix, which only contains K0 bases, and K0 is usually smaller than K. After we get this selected base weight matrix, we can use it for informative gene selection. So this is very intuitive. We only need to select those genes with the largest weights in the weight matrix. And another usage is for the new data projection. So you imagine you have a new data set. It can only has M genes as long as this M genes is in the selected weight matrix. And you can use the weight matrix to project your new data set onto the same low dimensional space as your original reference data set. And then you can use any machine learning algorithm to perform, for example, cell type prediction. Here is our result. First, we want to compare the weight matrix estimated from PCA and PNMF. In PCA, we can see there are both red and blue colors, which means that the weight can be both negative and positive. And also the sparsity of this matrix is very poor. But for the PNMF, we can see the sparsity is very high and all the weights are strictly non-negative. And we can see there is a very clear orthogonality between different bases. So the non-negative weights are almost not overlapping between different bases. This will greatly increase the interpretability of your data set. Therefore, we can use the PNMF as a functionally interpretable representation of the data set. Here we perform the Go annotation on the weight matrix from PCA and PNMF. We can see the Go annotation on PCA. Within one PC, there are a lot of different Go terms. And between different PCs, those Go terms are overlapping between each other. But for the PNMF, within each basis, almost all the Go terms, they are related to one specific functional group. And between different bases, the Go terms are almost non-overlapping between each other. Therefore, scPNMF itself can work as a very interpretable linear dimensionality reduction compared to PCA. We want to show why the basis selection is an essential step. Here we show 5 different bases and their correlation with the cell library size. And we can see some of the bases they are highly correlated with the cell library size, and we want to remove them. And also some of the bases, they only show the unimodal pattern, which means that they are not contributing to the underlying cell type structure. In order to validate this selection, here we use UMAP to show the genes from the selected bases and the unselected bases. Here we can see using the genes from the selected bases, we can perfectly distinguish the 3 cell types. But for the genes from the unselected bases, the cell types are just mixed together. Finally, we want to show the PNMF generates a better gene selection compared to the state-of-the-art methods. Here we compare scPNMF against 11 different gene selection methods on 7 different data sets and with 3 different clustering algorithms. Here we use the ARI as a metric of the clustering performance. So the brighter the color, the better the clustering results. And we can see scPNMF generates the best results across all data sets and all different clustering algorithms. We also use the UMAP visualization to show our better performance. And here on this data set, only scPNMF can perfectly distinguish the 4 cell types, while almost all the other methods, they fail to distinguish 2 similar cell types. We also benchmark these results with a large range of different gene numbers from 20 to 500. Here we can see the average ARI of scPNMF is always higher than other methods. And another interesting phenomenon here is that after 200 genes, almost all methods, their performance become quite stable. Therefore, these results might inspire us that 200 genes is perhaps a sufficient number for representing the major cell types for different single cell data sets. Eventually, we show how the scPNMF can be used for targeted gene profile. Here we have 2 reference data sets, JON8 and PDMC, and we use it to generate the pseudo-targeted gene data set with only the selected genes. And we use 3 different prediction algorithms for cell type prediction. Again, we still compare scPNMF to the 11 different state-of-the-art gene selection methods. And we can see scPNMF leads to the best cell type prediction accuracy. Therefore, scPNMF can be very useful for targeted gene profiling experiment design. Here is a conclusion. So scPNMF is an unsupervised method for sRNA-seq data. It has 3 different major applications. The first one is informative gene selection. So it can let you specify a small number of genes, and the gene selection method is better than the state-of-the-art gene selection method. And second, it can help you to map your new cells to the reference. So imagine if you measure the targeted gene profiling dataset with only the selected genes, and you can use the scPNMF to project your new cells to the low dimensional space of the reference dataset, and you can perform the label transferring. And eventually, finally, it can serve as a dimensionality reduction method, and its interpretability is much higher than other linear dimensionality reduction methods. In one word, if you have a situation that you want to use PCA or NMF, you can always try PNMF as an alternative. Finally, I want to acknowledge my collaborator and also the co-first author of this paper, Ke-Xin Li, and my supervisor, Dr. Jingyi Jessica Li. And we also want to thank Dr. Roy Woman and Zach, and also all the other members of GSB lab. Here is a link of our paper, and here is a link of our package on GitHub. We are very welcome for any questions and comments of our package and the algorithm. And thanks for your listening today.